So it's also my first time in Mifobio, but like the three over the previous speakers, I have no excuse, I think, because I share the animation of a research team at the Laboratoire Photonique Numérique et Nanosciences for almost 10 years with Gaël Rocher, who is a pillar of nanobio, of Mifobio, and she basically asked me almost every day to come to Mifobio. I always refused, find excuses. And finally, Olivier and Cécile did convince me, probably because, I mean, I felt they were desperately searching for a cheap speaker. But, okay, so thanks to them, but I really can say that I don't regret being here, and I will be back in the next years. Timo this morning talked about the physics of lots of diseases like dystrophy and cancer progression, evolution, and so on. Beautiful physics, but I would like to say that instead I am interested, and my research is dedicated to life instead of disease and death. But not the life and that as many others and very famous guy that we all know, love, admire, like Elon Musk or Jeff Bezos, because they all are oriented and they invest a lot of billions of dollars in trying to look for eternity, for basically prolonging. They claim they will prolong the life expectancy up to 150 years within a couple of decades. But I'm still interested in life, in the beginning of life. And actually, so at the beginning, this tree of life that we call, there are totiponent cells that will quickly become extra-embryonic materials and pluripotent stem cells. These pluripotent stem cells, as you all know, probably have the capability of self-renewal and also to give rise to all the cells that you find in our body. But so they will differentiate and get ramified, and for example, and at an intermediate level, they will give rise to all type of progenitors, like cardiac progenitors, endothelial, keratinocyte, and so on. But also, I put it in green here, to mesenchymal stem cells. And mesenchymal stem cells are already committed, but they will finally give rise to differentiated cells, but only a limited numbers of them, like osteoblasts, chondrocytes for cartilage, muscle cells, and so on. So, I want to mention here, because we use that afterwards, the most, I think, to me, fascinating Nobel Prize in medicine in 2012, but the discovery dates back from 2007, by Shinya Yamanaka, who was able to take differentiated somatic cells and to reprogram them into pluripotent stem cells, the ones that are basically at the origin of life. So these are called induced pluripotent stem cells, and it is just done through, by adding 4 transcription factors. These are called the Yamanaka factors. So it's really just as if you make a jump back in the future. But I like to say that because let's suppose I take skin from my, cells from my skin, a 55-year-old guy, and I am able to reprogram them into embryonic pluripotent stem cells of myself 55 years back. So you can make like this. It's a special dédicace to Olivier, who is a fan of Austin Powers. You can make a lot of mini-me like this. If you are, in principle, if you have reprogrammed some of your cells into pluripotent stem cells. So, but I also understood that, okay, we are in module 5 I have been recruited as a speaker of module 5, so we have to talk about mechanotransduction. And so I make the link with mechanotransduction and stem cells. As you all know, cells receive mechanical cues from the outside world, and they convert it into a chemical signal that may change the cell fate, and through mostly, I mean, either directly or indirectly, deformation, activation, regulation of transcription factor in the nuclei. So cell fate can be changed. It can lead to apoptosis, proliferation, increase in migration, contractility. But I'm really interested, and I was quite obsessed since for like the 4 or 5 years in differentiation. And the most remarkable example that we all know is the work by Dennis Disher. By the way, Dennis Disher also did a postdoc with Evan Evans a few years before me, and but he has the reputation of being very tough with the students. I'm not. I think. I hope. I don't know, but I'm not as tough as him. But I really. I mean, I love everything he does. I respect him a lot, and he worked on mesenchymal stem cells, so MSCs. And shows that they are basically chameleons. So that means that when he plates them on a substrate that is very stiff, they become, they start to express osteogenic genes and become bone-like cells. If he plates them on soft substrate, so mostly it has been shown that the YAP factor is inhibited, is inactivated, remains cytoplasmic, and that gives rise to neuron-like cells. I say neuron-like, osteogenic-like, because if you look carefully, the differentiation is not completed. If you want to complete it, you have to add now soluble factors. But actually, elasticity of the matrix, or more generally viscoelasticity of the matrix, is able to trigger, to make cells commit into towards differentiation. And actually, since then, you can find that if you look at the literature, there were, since like 2007, 8, after the discovery of iPSCs, there are lots of reviews claiming, trying to summarize the mechanotransduction of stem cells and of stem cells in general, right? But actually, if you look more carefully, and if you actually read the papers, there are also some papers saying that, for example, soft microenvironment promote the early neurogenic differentiation. So if you look carefully and you don't, beyond the title, if you really read the papers, you can see that, for example, on this case, when iPSCs are seeded on a soft substrate, it doesn't actually trigger, the differentiation is not triggered into neurons, but actually it's enhanced. So basically, you always need soluble factors. I couldn't find one paper in which experiments have been done without soluble factors. There is only a potentiation, a promotion, an enhancement, but in the presence of soluble factors. And there are even more contradictory and surprising results, like this one, where you can, if you apply a cyclic stretch on them, there is not a differentiation, but actually a reprogrammation. So that means that you come back to the cells that you created your cell line of iPS before. And for those who are not familiar actually with iPS, you may know or may not know that actually when you seed cells on a iPSC or embryonic stem cells in general, so pluripotent stem cells, if you seed them on a substrate, you immediately see colonies, but at the peripheries of these colonies, there are differentiated cells that really show up. And basically, there is still now protocols that are published in 2023 in order to really select the cells that have not been differentiated. And the protocol is the same that has been used. That has been used 15 years ago. It's this one. So you take a microscope and a marker, and you come underneath. You repair. You detect actually. You visualize the colonies, and you repair and you mark the colonies that are differentiated, or the packs of cells that are differentiated, and the ones that are not, in order to remove them afterwards with a pipetter or something like that. So pluripotent stem cells spontaneously and in an uncontrolled manner differentiate in 2D cultures. But if you look at embryo development, you can find out from when you go from a zygote to a gastrulation, let's say, you can find out that there is a special period that is about between implantation of the blastocyst on the uterine wall to gastrulation, in which embryonic stem cells are conserved. They form an epiblast first, and actually they maintain their pluripotency. So you can think, okay, that's because there are lots of soluble factors that are present and indeed avoid differentiation, because we are waiting for lots of morphogenetic changes before that. I can say that because indeed. So if you look at this movie, indeed, for example, if you look at hatching of blastocyst, you will see, if you look at the one at the top, there is a blastocyst cavity that is forming, expanding, it will pulse a bit, and then it gets out of the zona pellucida that is hardly visible here, just a bit dotty here, for example. So it's completely squeezed through the zona pellucida that is breaking. Right? So there are mechanical stress, there are compression, squeezing, and so on, but there is no differentiation at all. So, okay, as I said, there could be some soluble factors. So what people have done is have used, mostly iPSCs, and have tried to make some models, in vitro models of epiblasts without soluble factors. There are lots of work. I've just cited two, but each time, every time, either you embed iPSCs or embryonic stem cells in a matrix like matrigel, or you put them on top, and you obtain what people call epiblast models, or what I call cysts, because they are like luminized spheroids, hollow spheroids of cells. Right, and that clearly, and remain for a long time actually, pluripotent. So NANOG, OX4, and SOX2 are the three main markers for trying to look at the stemness and pluripotency. So the question we ask is, why do the pluripotent stem cells in 3D cysts maintain pluripotency, while they don't when they are actually in 2D monolayers, where they differentiate completely in an uncontrolled manner? So you can say, because there is no edge, right? Because I told you that they differentiate at the edge of the cell. There is less cohesion. There is, the tension, the membrane tension is a bit lower. it can spread more and start to differentiate at the periphery. But actually, I would like to show you that it's not so simple, and I'd like to make a kind of mechanical study of this puzzle without doing a lot of biology. Actually, not at all. So, just first, I want to remind you maybe of what are the different types of epithelium. We characterize the epithelia with basically the ratio of the height of the cell to the characteristic length on the basal side, for example. So there are some epithelia that can be very flat. They are named squamous. Some others have a height similar to the transverse dimension, but they are cuboidal. If they become very elongated, they become columnar. And there is also another one that may look like stratified, but it's not. It's a pseudo-stratified epithelium. So they defend, and they are found, all these types are found in different epithelia. So you can have a feeling that they depend on the cell type. Jean-François Joanny, Jacques Pro, Edouard Anelzo proposed almost 10 years ago that this epithelium, this cell morphology in epithelia is a kind of interplay between three parameters, which are, it's quite logical, the cell matrix adhesion, the cell-cell adhesion, and the contractility at the apical side. So if you increase the cell ECM adhesion, you can see that, of course, the cell will basically flatten, and you will get you will try to make what? Ah, yes. Okay. But okay, so it will give rise to squamous epithelia. If you reinforce the cell-cell adhesion, it will give rise to columnar, something like that. Hein? Ouais, mais j'arrive pas. So there is a beautiful work that Viana et al. 2 years ago that really make a nice morphologic analysis of a monolayer of IPS doing confocal microscopy, and you can see that it's quite flat. It's a squamous epithelium, and you can see also that cells at the periphery are slightly different. They start to differentiate, and you can also remark that it's a particularity of stem cells, IPS, pluripotent stem cells, that they have almost no nucleus, no cytoplasm. The ratio of, in terms of volume of nucleus to cytoplasm, is very large. And again, I show you the same pictures here in epiblast model in cyst. Clearly, it's not, the epithelium is not squamous. It's more like columnar or cuboidal. So we tried actually in the lab. Now I have set the questions, basically the puzzle that we want to solve. We have developed in the last years in the lab a technique that allows us to encapsulate cells. So we call it the cellular capsule technology, and it consists indeed of a microfluidic system. So basically you take three capillaries that are concentric, co-aligned. In the outer capillary, you inject a solution of alginate, very viscous solution. And alginate has the peculiarity of forming a gel, a porous and permeable gel, when it's in contact with calcium ions. In the inner capillary, you inject the solution of the suspension of cells. And in the intermediate one, you inject water, because it basically only serves as a barrier against diffusion. When you put cells in suspension, they have a tendency to release all their calcium stores. And if you put calcium in direct contact with alginate, it will plug the chip. So when you do this, you expect to form droplets, and then by falling in the calcium bath, the alginate will cross-link, and that will entrap the cells that are inside this droplet of liquid. So here we started with the system on the top that is full of glue, and then we switched to a kind of 3D printed system. And actually now the company that we have created is Tree Frog Therapeutics as a kind of all robot, the same size as Timo and my width. But it's but actually it doesn't work so well. It has just the advantage of being GMP certified. So when you do all this, you inject all the three fluids together. You get this kind of things. So it's taken with a high-speed camera. You get dripping, and the droplets have a size which is about 3 millimeters. Okay, I don't know if you, yeah. So, but 3 millimeters is actually too big because, I mean, all the cells will be in the core after proliferation, will lack nutrients and oxygen. Because in the body, I have always a kind of rule of thumb, in your body there are capillary vessels that span the whole body, that are exhibit a fractal structure, and that allow each cell to be fed with nutrient and oxygen. So basically, if you are there is not a single cell in your body that is farther than 200, 300 microns from a vessel. So we had to make droplets that are smaller than 200 microns. So to do that, so yeah, you could see before that it has nothing to do with the size of the nozzle. It's always 3 millimeters, which is the interplay between surface tension and gravity. But if you increase the flow rate, then you will generate a liquid cylinder. But this liquid cylinder has the peculiarity of undergoing a stability. It's an hydrodynamic stability that has been discovered by Rayleigh and Plateau and in the 19th century. And actually, the droplets that are formed have exactly, roughly, twice the size of the nozzle. So you can make like this droplets that are 200 micron in diameter. But you can ask then, okay, these are liquid droplets, and when they fall in the calcium bath, maybe they will completely smash and be destroyed. So we did that already also, and we looked with a high-speed camera, and you see that the droplets are significantly, because of the kinetic energy and the impact, they are significantly deformed. But actually, it's very elastic, because then they recover the spherical shape. I can show you how it goes in practice. So this is a nozzle. Here you can see in a beaker all capsules which are, there's a slight change in index of refraction, so you can guess them. And so when you look at it, at the bottom of the beaker, you see this carpet of capsules, which are indeed, if you do a confocal microscopy, which are indeed perfectly hollow. And if you look even more carefully, so they are not quite spherical. You can see that there is always this tail, which is due to the impact and instability, Rayleigh instability. And they are filled in average of, in this case here, about 20 cells per capsule. So you let them grow, you put them back in an incubator, and they can give rise to a spheroid. If we take here cancer cells, it was before being interested in life. I was also working on cancer at the Institut Curie. So you form spheroids that fill the capsule, so you end up with lots of spheroids that at the end of the day. There are also nice things. For example, this one is already confluent, it's full of cells, and look carefully, yeah, it bursts, and then the cells spread all over the Petri dish. And if you look more carefully, the capsule is coming back to its initial size. So that means that the material, alginate, is very elastic. So we use this, yeah, it was a work with Daniela Wigniewicz, and we use that for a long time to probe the pressure that was exerted by cancer cells due to proliferation. And because we use these sensors basically as, we use these capsules as pressure sensors. But that's yeah, like this. Sorry, I didn't. So if you monitor the deformation, and Timo said that, okay, a spring, it's like force equal to spring constant time deformation. And here we have a spherical spring, basically. So the force is the delta P, the pressure, and the deformation is this term U over R. And the spring constant can be calculated, and it depends on the material properties of the alginate and also the aspect ratio. But it's very straightforward. But now I go to IPS. So we did the same thing, try to encapsulate IPSCs and so I run the movie, and you can see that immediately you form a lumen that grows, the cyst grows actually inside the capsule. Okay. So you can take snapshots, try to track and to derive the cell layer volume as a function of time, from which you can derive the proliferation time of iPSCs. And actually, it's one of the fastest proliferating cells that I've never seen. 15 hours of basically proliferation time. And what you observe that is quite surprising is that the cell, the epithelium actually gets thicker. It's quite counterintuitive. The thickness goes linearly, goes linearly with the radius. This is actually, this has never been observed, because what happens if you consider all kind of other cysts that we know, like MDCK, that are very well studied, MDCK cells, some like for acines, is that what people say, and that's true, is that there is a secretion, lots of secretion in the lumen. If you create a high concentration of molecules in the lumen, you generate osmotic pressure that will also contribute to make the epithelium thinner. So usually you get, when you get bigger, you get thinner. Here, when you get bigger, you get thicker. And so how does that behave when you compress them, for example? And we have a nice way, I told you, these capsules can serve as pressure sensors. So if you let the system develop and reach confluence, the cells will be then compressed by the capsules. So so again, you can see the lumen, then it fills up the capsule, will fill up the capsule. And then we basically fix them, we stain them, and they are still perfectly pluripotent. by just staining with OCT4 and SOX2. You can see after 4 days, so we of course left time to see the effect of a possible loss of preponderancy, but no, we recover 97% of basically stemness. So if you look at the different days, you can see that indeed we start as soon as a lumen is formed with a cuboidal morphology, and then we go to a columnar morphology, and even to a pseudo-stratified morphology. Pseudo-stratified, we can think that it's stratified, it's actually not. You can really check that there is, I mean, the membrane of one cell is going from the apical side to the basal side. And so I told you, if there is osmotic pressure, it should get thinner. So we try to measure here the osmotic pressure, the luminal pressure. So to do that, we sort of glued through the alginate the capsule on substrate, and then with a pipette, we poke into both the capsule and the epithelium. So that's what's been done here. So if there was if there was a leak either at the capsules, on the capsule part or on the epithelium part, when you puncture with a pipette, there will be a deflation of the balloon here. There is no. So if there was a high luminal pressure, the balloon will also deflate. Why? It can be intuitive. It can be also explained a bit more quantitatively. If you put a channel, if there is a pressure at one side, it will induce a flow on the other side, which depends on the geometric parameters of the channel, and basically the pressure is proportional to a so-called hydraulic resistance to the flow. We have measured the decrease in volume. There is, in all cases, actually just note that it's a very tiny volume, less than 0.5 nanoliters that are involved in the lumen. There is a very little decrease in the lumen volume, and from that we can derive the lumen pressure that is of the order of less than 10 Pascal. So 10 Pascal is 1. So Daria before talked about 100 of kilopascal, and 10 Pascal is about 1 millimeter of hydrostatic pressure. So you can consider that they are basically not pressurized. Then we wanted to study the mechanical properties of these objects, and we didn't get, help by an artist like David this morning, a Japanese artist, but we got inspired from a physiologist, an Australian physiologist. He's in Australia, yeah. Yeah, an Australian physiologist, and also a poet. He wrote poems about the work in the lab. He was absolutely, a bit crazy. And he was also studying the elasticity of rubber balloons, of party balloons, and of bladders of cats, pig, dogs, and so on. So, but we did the same experiment. What he did is just he just added and injected water inside the bladder, and he measured the properties of the bladder. So we did the same experiment, and we used the device that I showed you before, but 100 times smaller. So again, this whole experiment that you can also inject liquid, and then the idea is to inflate the balloon. So that's Hanirban who did this. You need to have really kind of golden hands to do that. It's not easy, and also it's not easy to make a lot of experiments. So the idea is the following. in order to extract the mechanical properties. So I show you that if you have a difference of pressure inside an open-ended channel, the flow will be proportional to the pressure. Now, if you have a dead end channel, that's quite intuitive again, the flow will go backwards, right? Now, if you put a kind of elastic balloon, party balloon at one end, so if you apply a pressure, you will induce a flow that will inflate the balloon, that will increase the tension of the balloon, which will counterbalance the flow of the liquid. So that means that even though the pressure is increasing, you expect that when this pressure inside the balloon will reach this pressure that you have applied, there will be no flow. PA minus P is equal to zero. So the flow is equal to zero, and you reach a stationary state, a steady state. So we did these experiments, and I can show you here that when you apply the 1 kilopascal pressure, you see that the balloon is increasing. Now you see it's going slower and slower, but actually there is no steady state. We couldn't find any steady state. But so if you really want to derive anyway a kind of elastic modulus, you can derive the one that is just at the beginning of inflation. So by doing a very simple model of like for an elastic system, so all notations are dimensionless analysis. So basically, from that curve, the slope of this curve, you can derive a Young's modulus that is of the order of 4 kilopascal for this system. But the fact that you don't reach a steady state means that at every step, the Young's modulus is decreasing. So if we did that at any steps, by measuring the lumen pressure as a function of the strain, the deformation, and we can go up to 80% deformation, you can see that basically the slope is smaller and smaller. That means that there is a strain softening. The effective Young's modulus elasticity of a cyst is decreasing. So generally, I think you can say, okay, that's a signature of plasticity. But we went on and we assessed for reversibility. Actually, if you inflate it and deflate it through different cycles, you can see that it perfectly, almost perfectly, goes back to the initial size. So it's reversible. Of course, as I told you, at the first cycle, you go quite high, let's say, but actually the modulus now has been decreased. So at the second cycle, you will inflate it more, and so on. But the important point is that it's reversible. And again, I can show you that here we applied a ramp of pressure, but by applying a ramp of pressure, we can reach, okay, it's more impressive if you convert it in aerial strain instead of linear strain, and we reach 400% deformation. So how to explain that? First, that they are super deformable, they are not plastic, it's reversible, they are purely elastic here. So a few years ago, Xavier Trepa proposed that there was a kind of super elasticity on a different system with few cells getting super stretch. But here we have a different mechanism. So I will not focus on Trepa's. So you can see here again that we did that under the confocal microscope with all the microinjector around on a live cyst. You can see here it's the pipette that is not fluorescent, and we inflate it. So the membrane has been stained here, I don't know if you notice anything special. What you must know is, so a cell is uncompressible. There are 3 different modes of deformation of a system by conserving the volume. First mode that you want to avoid is just to compress it, because you don't want basically to touch, to deform too much the nucleus. The second mode is if you want to keep volume constant but deform the cells, make it flatter, you can tilt it. And the third mode is it's a buckling, it's a bending. By taking that the cell is an hexagon in 3D, if you press on it, you will bend, you will buckle the edges. So bend and tilt, and we assess, and I don't know now if you look at it now, you can see that there are, for example, here, this one is buckled. I should have, so this one buckles, and there are other ones that just remain straight and, remain straight and tilt. See this one, for example, this one, I think. So Anirban really analyzed all this and tried to go beyond doing it manually. And we can associate an energy to the bending and to the tilting. I will go on that. So you can see easily that in a normal, in a resting case, most of the cells are radially oriented, and then some of them are tilted, and also the edges here are relatively straight, while the bend is more pronounced when they are stretched. So we did also, with the help of Madhavan in Harvard and Justin Tauber, who was a fantastic postdoc of him, some simulations. So, and he built a kind of 2D models with just sticks that can bend and tilt, and actually then run the simulation. If you look only at buckles, if you only consider a buckle as a soft mode, buckling, and then tilting, and then if you put everything and you introduce a bit of disorder in the system, and basically we compare that to our experimental data, and we think it matches quite well. So I'm almost done. The idea that we propose is that through these 2 soft modes, buckling and tilting, you can you provide a kind of mechanical buffering. Because if you don't have these modes, you need to compress the layer. And so, but the energy that is required if you involve these 2 modes, buckling and tilting, is considerably reduced. So the message here that I show you again, this movie, so then it bursts finally. But so the message here is that by looking also, it's not only because of the fact that the cysts have no edge compared to a 2D colony. It's not also only the fact that we are in a like spherical geometry, the cyst geometry. It really, you have to consider the epithelium, the morphology of the cells in the epithelium, and the idea of having, so the system is probably well tuned so that the cell adhesion is quite strong enough so that you have a columnar or a quite high aspect ratio of the cell inside, which basically prevent the nucleus to be too deformed. So the take-home message is that pluripotent stem cells are not mechanosensitive. So they don't respond to mechanical cues as MSCs are, as mesenchymal stem cells. And the idea is we have, but we need to investigate that a bit more, because there is, as you saw, there is no biology, just markers for stemness and mechanics. So we need to investigate more. But yeah, the point is they don't respond the same way. They don't respond. They don't trigger. Mechanical cues are not able to trigger the differentiation of pluripotent stem cells. And you need to start with soluble factors. That's a bit disappointing, but it's as is. And with that, I want to thank all the people from the BOF lab. So you can see Hanirban is just on the right here. There are other. So I said that we animate the team with Gaël, but also with Amaury that joined us recently. There is also Laetitia that you all know, because with Adeline, on the behind here, they are a lot involved in the cell culture room here at Mifobio. And also Laetitia is leading a platform, Bordeaux platform at the university dedicated to encapsulation. And we have, of course, a lot of interaction with Tree Frog Therapeutics that is dedicated to cell therapy and amplification of stem cells. And with that, I'm done. Thank you very much.
Thanks a lot. Very, very cool stuff, especially like punching in and blowing it up. This is great. But I'm not sure if I got the physics right, because like you assume that the elastic modulus stays the same, but isn't it expected?
No, it's not.
No, I mean, in the first idea when you said it would saturate, right?
Yeah.
Because
no, it doesn't saturate.
No, in the experiments,
yeah.
But in the theory that you wrote down. But in that theory, you said that the elastic modulus is constant.
Yeah.
But I think it wouldn't, because they thin out, and so the elastic modulus should be kind of like dependent on the thickness. Shouldn't it also thin out automatically just because they get thinner?
So we consider a thick shell, right?
But you have
the
thickness was in there.
The thickness is indirectly there, because there is air in and air out. So it's a thick shell model.
Ah, okay. So that
So basically, you have an effective elastic modulus that depends on the.
Okay. Yeah. Right. Because it thins out, and by this, like, if you would have one shell, it would actually.
So it's. I mean, the model is very simple, but it's quite. It's a kind of viscoelastic model if you look, but the effective viscosity is in is encoded by the hydraulic resistance. It's weird. But you can figure out, like, there are three lines of calculation, and it's but it's purely elastic. But the modulus is dependent on thickness.
And just to be fair, also, like, you showed that the differentiation was not as, like, they didn't differentiate in your system. But did you really look at the same amount of cells in the end? Because, I mean, here you just had some of these guys. Because you said before there were islands that were not differentiating.
But they do differentiate at the periphery.
No, but I mean, in your capsules, they don't, right?
Yeah.
But did you really get the same amount of cells to test that? Just to be fair?
The same amount of what, compared to what?
Of total cells. When you make a measurement in the 2D situation, right? Then you have seen that there are some of them are not committed?
Yeah, that's right. No, we haven't done it.
I mean, just to be, because it might be a probability question.
Right. Yeah.
Sorry. Because, like, I mean, it's great.
Yeah.
But
actually, on top of differentiation, there is lots of depth. So you can take depth as a kind of differentiation. But
you care about that.
But it's very difficult to count the number of dead cells because they are floating.
Yeah, true.
So you should accumulate depth and differentiation, I think, to be fair also.
Yeah, that is right. Okay. Thank you. Awesome.
So one question here. There. Right.
What?
Left. Super nice. Yeah, about soluble factors. I remember that something was important is actually in 2D that you need to provide the soluble factor from the basal side. And one of the mechanisms by which there was some kind of differentiation happening was the fact that the cells were not getting anymore the...
That we don't put soluble factors in our experiments.
No, but I do secrete maybe some of them, no? In this
but it would remain inside.
Yeah. I don't know, is it tight? I mean, there is no...
Yeah, it's completely impermeable. Even if you stretch it by 400%, it remains impermeable just before it bursts, or the defect.
Okay, and you haven't looked at what's.
But yeah, I see your point still. I mean, you would like to make 2D colonies on a substrate and
yeah,
feed.
And they did that actually when they take colonies, 2D colonies, and you feed them with a membrane from the bottom with BMP, or I forgot which kind of soluble factor. They keep. They stay.
We haven't done it. Yeah, that's a good idea.
No, it does. It's all.
Yeah, have you done it? That? No. Huh?
Not me, but people did it, and they
yeah, Sijia, okay.
Yeah, like in Sijia's lab, like people following their work. I think so.
When they do but they introduce really the soluble factor to induce differentiation.
No, to keep stemness.
Ah, really?
Yeah.
But I don't know then.
I think it's one of it's Fred Etok who did that. Like we can discuss that later if you want.
Okay.
All right, if there are no more questions, let's thank our speakers. Oh, one quick one here, sorry.
Yeah, I had a question. So when they go out of the alginate kind of structure, we see that you have some kind of EMT. So they escape from
when
the
they
aggregate.
Go out, in which case?
Like when they reach the size they have within, and then they start going out. So is it linked to the pressure?
You go from 2D from 3D to 2D, basically, and the cover slip is adhesive, basically, so they spread like.
But we even start to see that they go out when the pressure gets built up in the capsule, no?
Built up because of proliferation. It's just an increase in volume. Then the capsule is not indefinitely expansible, so it bursts, and then they have the freedom to spread everywhere, and mostly on the substrate. But it's you could say it's I don't know. I don't know if it's an EMT, especially in cancer.